This is a sort of introduction to quite a diverse bunch of species of wasps, which, although they're diverse, all happen to look much the same. So I guess this will be trying to illuminate, what these things are actually doing out there, how you might tell them apart from each other, but really it's a sort of introduction to the subject. So, nocturnal ichneumanoids, by the end, hopefully you'll agree they're amazing, but also you'll go away and find out the resources that will tell you actually all the details about them. This is more of a broad brush overview. This first slide here shows 4 entirely different species of Caspialus, typical nocturnal ichneumanoid wasps. You can see they look entirely different. No, they all look very similar. This is the sort of nocturnal morphology of wasps. As we'll go through the presentation, you'll see lots of wasps that look much the same. And one of the features about these wasps that fly at night looking for caterpillars is they tend to have long legs, long antennae, kind of skinny bodies, big eyes. So let's have a look. There you go. That's what might, if you're a caterpillar at sort of feeding happily on vegetation at night, you may suddenly be confronted by this vision of an ichneumoniid wasp pouncing on you and stinging you. That's what they do. They fly around at night looking for mostly caterpillars, sometimes sawfly larvae that feed at night. So you can see massive eyes for trying to gather light, including these big ocelli at the top. 1, 2, 3 big ocelli, which are helping to gather as much light as possible. So there you go. Oh yeah, I should mention now as well, this often comes up, debate as to whether they can sting you or not. Any of these wasps that are big enough to sting you can sting you, and I've been stung by loads of them, but it's not that bad. Generally, it's sort of like a fleeting pain, I would say. A bit like a common wasp, but then it goes after a few seconds. If you're a caterpillar, the effects are slightly more serious. You may end up dead. So anyway, before we get on to the sort of grisly biology of ichneumanoid wasps, what are these things? This is an overview of the sort of Uk Hymenoptera. So that's the insect order that includes ants, bees, wasps, sawflies, and of course, my lovely parasitoid wasps. And you can see a sort of pie chart splitting up the 7,700 odd British species of Hymenoptera. There's a lot of interest in the aculeates, that's a stinging ants, bees, wasps. Relatively small proportion of the UK fauna. There's a growing interest in sawflies, which again, quite a small proportion of the UK fauna, but really interesting group of Hymenoptera, and it's fantastic to see a lot more interest in those these days, actually. But still, this big slice of the pie are the parasitoid wasps. These are various groups that used to be called the Parasitica. I don't like to use that term. But anyway, these are wasps that go about that have a life history that involves eating another insect or another arthropod alive. And that's a huge proportion of the UK fauna. So it's about 6,500 species of parasitoid wasps just in the UK. Big group, not a lot of people studying them, sadly. There you go, there's some numbers. So of those 6,500 parasitoid wasps, a whopping 3,840 belong to just one superfamily. That's the Ichneumonidia, my favorite group. So that's 2 families, Ichneumonidae and Braconidae. So it's 3,800 odd species that are all doing fantastically interesting things out there, but we know very little about most of them. And every year we're finding new species. I believe Tony Hunter at Liverpool has just found a new ichneumoniid on the Sefton coast, new one for Britain, that sort of thing. It's brilliant. It's a brilliant group for finding novel findings. You can find new species for Britain much more easily than you could for, say, beetles. The issue, of course, is identifying them, but we'll come on to that. Here's a few ichneumonids down here. Cool wasps, basically. But we're not going to be talking about 3,840 species in this webinar. We're going to narrow it down. But I suppose this is the obligatory plug for my book. This is a handbook that I co-authored with Mark Shaw and Mike Fitton. It's published in 2018. It's an introduction to ichneumoniid wasps, which covers sort of biology, accessing the literature, how to identify subfamilies, all that sort of thing. And it's a successor to this volume on Braconid wasps, published way back in 1991. So it took a fair amount of time to get around to publishing the companion volume, but they're both out there. And you can download the Braconid Wasp volume for free from the Royal Entomological Society website. But just to point out, these sort of introductory texts do exist. And if you're really lucky, you got a slice of the Ichneumoniid book as a cake. That was at the launch party. My wife made that cake. It's amazing. It's far tastier than the paper version. Anyway, just briefly going back to what parasitoids are. So all these Ichneumonids, all these wasps are parasitoids. So that's basically, hopefully you've come across these things before. These are wasps. There's also parasitoid flies, a few parasitoid beetles, but most of the parasitoids out there are wasps. And what they're doing is developing in or on other insects. Also, quite a lot of spiders get hit by parasitoid wasps. And what they do is typically introduce some venom that will either permanently paralyze the host, in which case the wasp is basically eating a bag of fresh meat. The host is paralyzed and kept fresh. They're idiobionts. Horrible term, I know, but it's a useful term for wasp biology. But there's also a sort of equal number of wasps which sort of temporarily paralyze their host. Paralysis wears off and the host insect goes about its business again, but with an egg of a parasitoid in or on it. And those are coenobionts. And that's really important in terms of wasp biology, because this means that the wasps can exploit host insects which may not be accessible at later stages of their life. So they're attacking them when they are accessible and waiting to complete their development, typically until the host has formed a sort of a pupation site. And we're seeing that in a minute in some of the slides that follow. The nocturnal groups that we're going to be talking about, these nocturnal parasitoids, are all coenobionts, which is because they're attacking hosts that come out at night. They're basically going for these caterpillars and sawflies that are trying to avoid predators, but there's no avoiding wasps. They're always out to get you if you're a caterpillar. Obviously, parasitoids, massively diverse group. We're going to narrow it down. The wasps that we're going to talk about are quite few in number. But anyway, here's an example, not one of my nocturnal wasps. I couldn't help, but I just really wanted to put this in. This is a gregarious endoparasitoid coenobiont wasp. It's Cotesia glomerata, which attacks large white, or one of the cabbage white butterflies. This is the bit that makes some people queasy about, when it comes to wasps. They do eat their hosts alive. And here's a bunch of larvae bursting out. They're going to spin their cocoons, and that will be the end of the caterpillar. So there you go. Although there's a whole interesting biology going on here with Cotesia glomerata. That's for another talk. But let's just say that the caterpillar is not quite dead yet, and it still has some useful roles to fulfill, guarding the wasps that have just eaten its insides. But like I say, that'll be an exciting other talk. Before we go on, so basically that's the sort of biology. They're these little wasp larvae eating the insides of, in this case, caterpillars. So they're mostly feeding on the hemolymph and the fat. But things like that Cotesia, then leave it at that. These sorts of wasps, these Ophion, what they do later is after a while they end up eating all of the inside of the caterpillar. They finish it off. There's nothing much left. But they start off life as a wasp larva inside a living caterpillar, which is exciting. Just to point out, this is my website. It's pretty, I won't make any great claims about it. It's a very sketchy website, but there are links to literature from this, and there are some keys for identifying nocturnal ichneumanoid wasps. So for further details, do go to there, and so I'll be developing that as I over the next few months. So please do get on. Do contact me if you think if perhaps you've got interesting photos to add to it. That's always much appreciated. Or if you've got constructive feedback or telling me stuff I really should be including in this website, that'll be much appreciated. Basically, if you any spur to develop it is much appreciated, because otherwise I tend to forget and it languishes. But there you go. Have a look at that. Briefly, these two families of nocturnal wasps, we're going to talk about Braconids and Ichinomonids, these two massive families. They can be hard to tell apart, but fortunately, most of the nocturnal species are relatively straightforward. If you can see this Braconid wasp at the top, it has this cross vein, RS plus M. Ichneumoniid wasps lack that cross vein. So you've got a big discursive marginal cell as well. And they also have that vein 2 MCU, which we'll come on to in a minute. Basically on wing, so just looking at the wings, you can instantly tell apart all of these nocturnal Braconids and Ichinomonids. But there are other features as well, which help to tell them apart. This is some classic nocturnal Braconids. We've got a Zelle albiditarsus here. It's big. For Braconid, it's relatively large. And again, like lots of nocturnal species, you can see it's basically sharing, it has the same sort of color pattern as loads of nocturnals, which I should have said earlier. Most of these wasps that we're going to be talking about are basically a sort of uniform pale orangey red color, testaceous, I often refer to it. It's probably because if you're flying at night, there's no need for particular pigments. So it's cheap, it's energetically cheaper to just not lay down those complex melanins. So basically they're fairly uniform colors. You can see big eyes, long antennae, all the better for seeing and for sniffing out hosts at night. So here's two classic nocturnal Braconids. I put this Rogas in as a lovely example. This, let's go back. Rogas luteus. I just thought I'd highlight the fact this is a parasitoid of the festoon, which is a fairly widespread moth in parts of southern England nowadays. But it went through a big population sort of bottleneck in the early 20th century. I think it became really quite rare. And we think it may have lost some of its parasitoids. So this Rogas luteus, which is a lovely Braconid wasp, is no longer known from Britain, if indeed it ever occurred here. But just over the channel, you can find it quite commonly attacking festoons. So it's one to look out for in this country. Hopefully, it's here to make use of this caterpillar, because it's important. Yeah, caterpillars are there as wasp food. You see the beautiful eye as well of the Rogas. A lot of these nocturnal species have really quite striking eyes in life. Yeah, but sometimes that fades in death though. So there you go. A couple more Braconids. In this talk, I'm going to concentrate mainly on Ichinomonids, by the way, partly because the taxonomy of Ichinomonids is in a much more sort of it's evolving a lot faster at the moment. I think we've had for many years a much better idea of what species of nocturnal Braconids we have in this country and how to identify them. So changes are relatively few. But this is a lovely, this is a nice example of change. This is Macrocentrist Flavus, which Mark Shaw was sent from, it was posted to him from Devon a couple of years ago, and that's new to Britain. So this is probably a good example of a species actually arriving in Britain very recently. They attack, probably, we don't know for this particular species, but they probably attack sort of stem or leaf rolling tortricid or pyralid moths. And Macrocentrus has been quite well studied in the past. So when a fairly distinctive new species turns up in Britain, there's a good chance that we're actually finding, actually detecting colonization happening, which is quite cool. Here's a euphrine. So Pygostolus is a rather different sort of nocturnal wasp. As most of these things are going for caterpillars or sometimes sawfly larvae, Pygostolus attack adult weevils. It's a really weird biology. They actually sort of grab the weevil and they sort of slip their ovipositor, sticking out the end there, underneath the beetle and lay an egg into its abdomen. So, unusual biology. We've got 3 or 4 species in Britain of Pygostolus. But you can't really quite, I should have got a better photo, but obviously I've been working at home for the last almost a year, so limited chance to take nice photos. But just on the underside of Pygostolus, they've got this lovely sort of felt cushion. I guess it's something to do with grasping onto a weevil, but it looks very comfortable. It's a nice feature for Pygostolus. One of the classic, sorry, I'm whizzing through this because there's so many wasps and so little time. But one of the classic genera of nocturnal braconids is this genus called Aliodes. Now, Aliodes, there's loads of species. Some of them fly at night, some of them don't, but a few have this classic nocturnal appearance, like Aliodes praetor. Now, this is increasing. This is one of these species that used to be thought of as being quite rare, but it turns up a lot more now, probably because there's a lot more light trapping, a lot more people taking note of the fact of these wasps that turn up in light traps. So Aliodes praetor is a parasitoid of hawk moth larvae. This is a mummified lime hawk moth. So Aliodes and other species of the same subfamily, Rogodini, have an amazing biology where they basically inject. So they live as endoparasitoids, the larvae. They're inside the host caterpillar. And then when they're ready to pupate and, well, finish their feeding and then pupate, the larva, the wasp larva, releases some kind of chemical that basically transforms the host caterpillar into a hardened mummy. So these mummified caterpillars, these mummies, are really quite distinctive. You can often see them on tree trunks, on high bits of vegetation, and it can be quite. Some species of Aliodes are identifiable on the basis just of the mummy that they have caused. So basically, another lovely feature of Aliodes and some other Rogodines is you can see that mouth, that face in the middle. It's got a big sort of circular opening, roughly circular opening above its mandibles. This is called cyclostome condition, and this cyclostome opening is just basically part of the face is sort of curved inwards and shiny. It's a really nice feature, and if you can see that, you can see that on a nocturnal wasp, you know it's going to be a Rogodine. So cool things. Rogodines, Aliodes are a lovely group because they attack so many different species of Lepidoptera, and basically you can rear them quite nicely in the lab. So my colleague Mark Shaw has been working on them for years, working out basically what hosts does a species of Aliodes use, what constrains that host usage, how did their host range evolve, that sort of thing. And with Kees van Achterberg, he's publishing these series of papers on Aliodes. Now, this is sort of touches upon one of the problems that is, although nocturnal ichneumonids are obviously fantastic and interesting, they do come with their problems, and not least is the sort of poorly developed taxonomy of some of these groups. So Mark and Kees have been, this is the first two papers on Aliodes, and there's at least another two to come. And in the first paper, they dealt with 27 species, and 7 of those were described as new. In the second paper, they dealt with 42 species and another 6 new ones. There's loads of new species to be described, and loads of different species, loads of different names that have been applied wrongly in the past. So yeah, it's an evolving, literature's evolving. Thankfully, most of the papers I'm going to be sharing in this are open access, like Zuki's and European Journal of Taxonomy. Anyway, so that's a quick whiz through the Braconids. We're going to go on to Ichneumonids now. I'm going to deal more with Ichneumonids, mainly because it's my speciality, but also because this is where there have been most interesting changes recently, I would say. Anyway, basically there's 2 major groups of Ichneumonids that fly at night. One of them is the genus Natilia on the right here, and the other major group is a subfamily Ophionini. Now, Ophionines are really easy to recognize when you've got with a little experience, because they have this characteristic, as I call it, the horsehead disco submarginal cell. So this big cell here is sort of elongated, a bit like a horsehead, and it goes beyond. This is a cross vein called 2MCU, and the cell reaches across that to way past that vein. Whereas in most other Ichneumonids, this disco submarginal cell ends before vein 2MCU. So that's, if you've got a big sort of pale orangey reddish Ichneumoniid at light, and it's got this characteristic cell on the left, then it's an Ophionine. So let's just look at a few different Ophionines. So you've got something like Ophion minutus. It's a classic quite small Ophion which flies in May and June in woodlands, where it attacks geometrid caterpillars. Just a couple of features that you need to, so obviously when you're getting to, if you're into identifying these things, you will need to get used to a whole load of different terminology compared to many other groups of insects, compared to many other groups of Hymenoptera. But this is the cell 2R and RS, which bends down. You can see in Ophion, it's just a nice gentle curve. In Enicospilus, it's sort of slightly more sinuous and it thickened a little bit. Eromatilis and Steropoctonus, you've got a sharp bend in that vein. It's kind of a useful feature. You've also got things like these sclerites popping up in some Ophionines. So you get this. So within Ophionine, you've got different sorts of wing venation, which can be really helpful in identifying the genera straight away. Just to point out, these are 3 of the 4 genera we have in Britain. The 4th one is Steropoctonus Bombisivorus. This is the only species in its genus in Europe. It's a very cool thing. It's not quite like the other nocturnal wasps that we'll be meeting, because it's got all these quite distinct black marks all over its body. And it does fly at night, where it attacks the lobster moth. So that's an interesting, so this particular wasp, Steropoctonus, was always thought to be very rare in Britain. There are very few old specimens. I'm seeing quite a few now, especially through social media and through iRecord. So people are finding these really spectacular wasps in their light traps and recording them. So it is turning out to be fairly widespread in southern Britain, but much more restricted than its host. Lobster moth is really quite a widespread moth. So presumably this wasp requires something more than just its host to be there. It requires it may be temperature dependent, it may be habitat specific. We just don't know with these wasps. There's an awful lot to find out. Anyway, so that's one to look out for. And if, it may be spreading, or it may just be that we're detecting populations by light trapping more. Time will tell. And let's go back to that genus Enicospilus. I've got quite a fondness for Enicospilus just because a few years ago, I decided I was going to write a quick paper with Mark Shaw, just summarizing the species of Enicospilus in Britain and roughly where they are and what they do. So what their hosts are and preliminary distribution maps. So it's going to be really nice and straightforward. And then I found out that most of our UK specimens have been misidentified. We had a couple of species new for Britain. 2 names had been misapplied. We had to describe a new species. It all became very complicated, which I suppose is quite fun if you're a taxonomist like me, but all adds several years to any project. Anyway, one of the interesting things about Enicospilus is they have these sclerites. Many species have these sclerites in the forewing, and these little bits of chitin which sort of look like they've drifted off from the wing veins and are drifting across the sea of wing. These are, in Britain, these are quite distinctive for the genus Enicospilus, but a few other tropical groups of wasps have these sclerites, and nobody really knows what they're for. There's some theories. One of the more outlandish theories I heard, which may be something in it, is that it may be something to do with confusing echolocation by bats. Who knows? But yeah, anybody got any great ideas, let me know. But the exact pattern of these sclerites can be really informative for identifying species, thankfully. And you can see here's a little species. So the one with the sclerites marked, proximal, central, and distal, that's a thing called Enicospilus Merdarius, which seems to be restricted to sand dunes or sandy coastal locations where it attacks sand dart and possibly some other species of moth. And down below that, we have Enicospilus vermigulus, which is really fairly common and widespread, where it attacking things like campion, lychnis, these hadonine moths mostly. Then upper right, we have Enicospilus repentinus, which we'll come on to in a bit, which is totally unknown biology. And then we have Enicospilus inflexus, which is by much bigger than the other species. And interestingly, it doesn't have any sclerites, but it does have a very kinked wing vein. Also, I love some of the names attached to bits of their bodies. The fenestra is an area of the wing, a window, an area of the wing lacking any setae or hairs. I like that. So, okay, so if you're identifying these things, it's one of the important, it is often quite difficult because they do look pretty similar. These are two species, Enicospilus inflexus and Enicospilus undulatus, which are some of our bigger ichneumonids. They're really quite impressively large, and that's because they're going for lasiocampid caterpillars, so eggers. You can see, but identifying these two species is really quite tricky. You need to be able to, ideally, you need to be able to look at the head from dorsal view. These blue lines sort of indicate the sort of fat head of undulatus compared to the narrowed head of inflexus. It's an important point because sometimes I get sent photos of somebody thinking maybe I've got an Enicospilus undulatus, and to be honest, it's really difficult from a photo to know whether you're truly seeing this difference, and I've never yet been able to verify one of those from a photo. It really is a specimen job. But that's the importance of our collections. You know, I've gone through, these are some very preliminary distribution maps. There's Enicospilus inflexus, found, obviously this is just a reflection of the collections I've looked at. It's found throughout Britain, basically, wherever you find things like oak egger and northern egger, possibly drinker moth, I'm not sure. But if you go through the collections, you then find, I found no recent specimens of Enicospilus undulatus. So these three dots represent the only British specimens. The latest one was from about 1970. So maybe this is extinct, who knows. It seems to be a parasitoid grass egger, which is not a species that's doing particularly well, but it's still here. Anyway, one to look out for. So again, these are on the website, but useful literature. The British species of Enicospilus, I thought we were being really quite smart in finding 9 species in Britain. Several of them were new. One was, well, new for Britain. One was newly described. Then my colleague Nicholas in Sweden found they had 11 species in Sweden and an additional 3 new species. So there's always things to look out for. Some of these could be in Britain as well. But two useful papers there. The other genus of, another genus of Ophioniinae is this rather obscure genus called Eromatilis. A lot of people don't ever see these. They tend to be quite localized. There's only 2 species in Britain. As you can see, this is without context, this looks like a blob of insect. This is the middle bit. It's the mesopleuron. It's the side of the thorax. And as you can see down the bottom, there's a tiny little projection, a little lump, and that's very distinctive for Eromatilis, together with some wing vein characters. But these are our 2 species are really poorly known. You've got Eromatilis curvinervis has been reared from brindled green, which again is quite a common moth, but the wasp only turns up every now and again. I've seen very few specimens. And Eromatilis marginatus was supposedly reared from the satellite, which of course is a very widespread moth. But the wasp itself is very restricted in Britain. Where it occurs and can be quite common. It's a lovely sort of darker reddy orange color with black patches on it, and sort of fairly yellowish, slightly yellowish wings. And I used to work at Monk's Wood in Cambridgeshire, and these things were quite common there, flying around the wood at night. So I got a bit blasé about them, and since then I've hardly seen a specimen. They're really restricted, so it does make me wonder whether they really are parasitoid of satellite. One of the issues with wasps in general is how much you can trust the literature. There's so many misidentifications and so many assumptions about biology which are difficult, which have not been proved. But that's a talk for another day. Before, something I should have said earlier on, okay, if you're trying to identify these things, what's the difference between males and females? Now, these are just 2 pictures of the ends of the abdomens of Ophion species. Now you can see, unlike So a lot of parasitoid wasps have quite long ovipositors for finding and for laying their eggs in hosts. These nocturnal groups, many of them have quite short ovipositors, and that's because they're going for exposed caterpillars. So basically they're sort of pouncing on a caterpillar and laying an egg in it. So you don't need a long ovipositor for that. So this, if you can see on the left there, this ovipositor has a little notch towards the tip. That's classic feature of endoparasitoids. It acts like, what it does is it acts like a bit of a clip. It shoves the ovipositor into the skin of the caterpillar, and the cuticle or skin of the caterpillar rests on that notch. So it sort of keeps it in place, injects some venom, temporarily paralyzes the caterpillar, and then squeezes an egg down that. So this notch is found in those species in which they're sort of jamming the ovipositor into the caterpillar long enough for it to lay an egg. In species which are ectoparasitoids, i.e. they're just laying an egg on outside of the caterpillar, they don't need a notch. So it's a really useful clue as to their biology. But the important point here is that the ovipositor is very short, so it's easy to miss. But the sheath, so at rest, it's hidden by these sheaths which poke up. They're quite narrow. There's the male Ophion on the right. The genitals, the sort of external parts of the genitals, I call them the parameres because I'm old-fashioned, or the claspers. These bits can often be quite large and they're quite wide. So if you can sort of see a narrow bit at the back as opposed to a wide bit, then you've got a female. Going on to this lovely genus Ophion or Ophion. These are really quite diverse in the sort of north temperate areas where we live. Down in the tropics, you find Caspialus is by far the most common genus, but up here you don't get many species. Instead, you've got loads of Ophion. Now, Ophion, again, loads of them look the same. So this is I wanted to spend a few slides just sort of looking at Ophion and how you go about identifying these things. But at the top here, there's Ophion Ventricosis, a fairly widespread spring species which attacks some geometric moths such as the lilac beauty. It has these darker patches on it, so it's relatively distinctive. Often flies by day, unlike most of the other Ophion. But below is a much more typical Ophion. It's Ophion slavosecai, which is one of 20, 30 species, which all look pretty much the same. And one of the things I'm doing at the moment is going through lots and lots of UK specimens of Ophion, trying to re-identify them, because this paper came out a couple of years ago by Nicholas Johansson and Björn Söderberg. And this Nicholas has really transformed sort of what we know about Ophion in Northern Europe. It's got 18 new species from Sweden and nearby countries, and quite a few of those are turning up in Britain. So massive change in what we know about Ophion. Just to show you long lists of boring names, but that's what we knew in 2016 when I wrote the checklist. That's our list of UK Ophion. 2021, we know quite a lot more. All these different species, and only one of them, we've actually got rid of one of the species from our list because it turns out to have been misidentified. But the point here is most of these species have been here all along, just not recognized. The importance of heads again, plate from Nicholas's paper. And we'll come on to one of the things I want to stress is the importance of, if you're so inclined to build up insect collections, it's really helpful to build up a good collection of Ophion before you start trying to identify them to species. Because some of the differences become really apparent if you have them side by side. But if you're just running a single specimen through a key, say for the first time, it's so easy to go wrong. So down at the bottom, you've got 3 heads side views. Now, with a bit of practice, the difference in the sort of the fatness or narrowness of the head behind the eye becomes quite distinctive. And again, the narrowness or wide width of the face, these are really important characters, but so subtle. Also, an important bit to look at in identifying Ophion and lots of other ichneumons is the Propodium. And for those who, if you've not come across a Propodium before, it's a weird feature of Hymenoptera. So it's the first abdominal segment, but in all these parasitoid wasps, this first abdominal segment is fused to the thorax. So it looks like the end of the thorax. And what looks like the first segment of the abdomen is actually the second segment. So if you can see that on this Ophion here, this skinny segment at the base of the abdomen, that's actually the second abdominal segment. It's a weird adaptation to moving your abdomen underneath so as to lay eggs in hapless insects. Yeah, if you're light trapping at the moment, you might come across this beautiful thing called Ophion Obscuratus. Well, we used to call it Ophion Obscuratus. I think it's the only Ophion on the wing at the moment, although look out for Scutularis. It's always been recognized that this can't be just one species because they have different generations. And as far as we know, each Ophion species in Britain only has one generation. They attack a host, lay an egg in it, and they have basically an annual cycle. So that larva eats the larva of the host and then pupates and pops out again the next year. But Obscuratus flies in several supposed generations through the year. So Nicholas Johansson has formally described some of these as new species, but some of them weren't dealt with by his paper. So by my reckoning, what we used to call Ophion Obscuratus is now a lovely 6 species in Britain. 4 of them seem to have names. So there you go. 4 of these are richly patterned, with these lovely sort of creamy marks all over the abdomen and the thorax. Whereas, and 2 of them are poorly marked. So there's an immediate way of telling those 2 groups of species apart. And now, interestingly, so the one that's on the wing now, sort of our late winter species, this doesn't seem to have a name, or at least not, I can't find one. So that may be an undescribed species. Then in spring, in early summer, you've got this Ophion variegatus comes out. That's quite common. There's possibly another thing called species 2 at the moment. I've seen one specimen. It's a weird, small, shiny thing. Sylvestris is out. Then in the autumn and early winter, right up until December, you've got autumnalis and obscuratus, the true obscuratus. So there's a lovely mix of things to get your eyes into. And again, if you've got some of these in your collection, it's be a great opportunity to start trying to sort them out. So I've just started, this is just to show, so with Ophion, I'm having to go back and start really looking at all the UK specimens that I can again. And these are some quick photos I took of the back of the head. This is a really useful character. This is called the occipital carina. Basically, it's a division between 2 segments that are fused into the head capsule. The point is this ridge runs down nice and smooth in the true obscuratus. This is a Ophion variegatus where it seems to have a bit of a wobble, this ridge. And my undescribed species, which is quite common, has a distinct bend there. So yeah, there are some nice morphological characters. But we're just starting to understand how to tell these species apart. And a lot of this comes down to counting segments of the antenna, which can be quite tedious. So here you go. I just went through my data set this morning, actually, and to see at different times of the year, does the average number of antennal segments vary? Yes, it does, although not entirely clearly. But you can see that the ones with long antennae come out in September and October. Then you seem to have some in January again. And now, these are very small sample sizes, by the way, so I'm sure this picture will change a bit. But the ones that are out now have quite short antennae. And again, the spring variegatus has quite short antennae, very short antennae in August. And this is roughly their flight times. So theoretically, you can identify the different species by knowing which generation or which cohort they are. It's not quite as straightforward as that, obviously, as you'll see, the peaks sort of merge into each other a bit. But basically, in the middle of summer, there's hardly any of the Ophion obscuratus group on the wing. You get this big emergence in October. That's when their obscuratus is quite common. Again, they tail off. Then you've got this late winter peak now. and I suspect they're not one of the, yeah, we'll come on to the poor quality of my data, but the relatively small number of specimens I've seen from February is probably not a reflection of how common they are. It's more the fact that there's not as much like moth trapping in February. Hopefully that will improve. And then you've got the spring form. So yeah, just to point out, you can sort of see some of these different species at different times of the year, but this is all very preliminary work. It's quite exciting if you're into the differences between very similar species. But what are they actually doing? Surely that's more interesting. This is Ophion slaviceki, which I used to call Ophion luteus until Nicholas pointed out it was a misidentification. But here's just a quick photo. This is a common species, flies in August, September, a little bit later sometimes. It's really quite ubiquitous, but for many years we didn't know what it was doing. And then I had a hunch it was attacking heart and dart, and I proved that by rearing some heart and dart caterpillars. This is a phone photo, so it's not great quality, but basically the wasp, this is when an Ophion larva burst out of the heart and dart caterpillar and started to spin its cocoon. So that was very exciting for me. You can see it's got a white silken cocoon. It takes several days to spin this. Eventually it hardens, melanizes into this dark brown tough cocoon, which helps it overwinter in moist soil. Really, that was quite good. I was really pleased with this host record. And then it turned out 2 other people had also reared it about the same at different times. So we collaborated and published that. Again, just the things you can find out by just looking for things that nobody's looked into before. Heart and dart, really common moth, but nobody bothers rearing the larvae. So if you do, you find things like, you can find out some interesting things. Just want to show you mandibles. Again, Ophion slaviceki is quite distinctive in that if you compare it to Ophion crassicornis, this one in the middle, Ophion crassicornis, is a typical Ophion mandible. It's quite big with an internal angle between the 2 teeth. Slaviceki has this, lacks the internal angle, and often you can see their mandibles are worn down, sometimes really blunt and the teeth are almost missing. And that's because what they're doing, as we found out, they're attacking heart and darts, and then they're the adults are having to dig their way out of the soil, probably by August, quite compacted, hard soil. So I guess they're wearing their mandibles down. So again, so that's quite, yeah, that's something we found out about really common species, and some of these species have never been reared at all. Just to mention, the Tanyptera, it's a little group of wasps, Ichneumoniid wasps, which attack sawfly larvae. There's 3 genera in Britain which you can quite commonly find: Absyrtus, Opheltes, and Perilissus. I won't go into detail. They're covered in my keys on the website, but they all have this really cool clypeus, kind of ridged and with little lobes. Clypeus is the lower part of the face. And they often have big, long, lower teeth. Don't know why. Mesocorini, another small well, this is quite a small subfamily of wasps. These are quite distinctive. The females have a very needle-like ovipositor. Males have these long, spiny parameres. No idea why. Biologically, this is a really interesting group. They're hyperparasitoids. So basically, they're flying around trying to find caterpillars that have already been parasitized. And then they'll lay their eggs inside the ichneumoniid or braconid larvae inside. Natalia is my true love in the wasp world. Natalia is quite a big genus of nocturnal wasps. About 25 species in Britain. You can see at the top, this is, you can imagine, head towards the right, sting towards the left. These are the first metasomal segments, second abdominal segment. Ophion have these really skinny base to the apparent abdomen. Natalia is thicker and they've got these big holes. Quite a nice distinction because they look very superficially very similar, but in detail, all the details their morphology are quite different because they're very distantly related. But here's some caterpillar death. Sorry about that. We've got shoulder striped wainscot on the left, and you can just about see 2 larvae on it. They've just hatched from the eggs. That's Natalia has laid eggs on the eggs are anchored into the skin of the caterpillar. The larvae have just hatched out and they're going to start feeding. And that's because the caterpillar has basically made a little hole for itself in the soil where it's going to pupate. The Natalia wait until the caterpillar is ready to pupate before they start eating. And on the right, this is just a picture from a website, but the pus, this is the pus moth. And it's being attacked by Natalia vinuli. There you go. These are Mark Shaw's lovely photos. Natalia vinuli laying its black eggs on the host, the pus moth, and then it will. What happens? Eventually, they wait until the moth has formed its cocoon, this tough cocoon on the right, and then they finish off the caterpillar, eat it quite quickly, and the wasp larvae spin their own black silken cocoons inside. Very cool things. Natalia are lovely. Biologically, they're really interesting because you can see them develop, obviously, as they're ectoparasitoids on the outside of the host. Also interesting because they've been very neglected. So, this is a new species. It doesn't have a name yet. I've got 3 specimens from Northern England. I've not seen it from anywhere else yet. We've got a few other species that need names as well. If you're identifying these things, you need to look at often subtle differences in the wing venation. So the veins, sort of the different sort of angles between veins. That's quite useful, or the gaps between veins. Also, the male genitalia are unusually useful in Natalia. They often have this pad inside. I'll show you that in a minute. Also, the Propodium, again, that first abdominal segment that's fused to the thorax. These complex systems of ridges and little grooves and things, they're very useful. So things to look at if you're looking at Natalia. This is another undescribed species. There's a whole bunch of Natalia species which all look really similar. They're very frustrating. You can see this one has got very narrow head. The temples are very narrowed behind the eyes. And there's no gap between the ocellus and the eye. Females in this group often have testaceous area between the ocelli. Males have it dark brown. There's several, in Europe, there's about 5 different species which look incredibly similar. We have 3 of them in Britain at least. This is a thing I'm calling Natalia williamsi, which turns out to be quite common in southern England, but had not been described. I've just started looking at claws, the details of the claws and how wavy these, this is the comb, the pectination on the claw, which they use to hold on to. This is a male, so they're using this to grip onto the female, but females have fewer teeth. But anyway, the differences in shape are really quite useful. Just had a go yesterday with a nice lens trying to capture the details of these claws, so I'm not quite there yet, but you get an idea. Yeah, male genitalia, just to point out, in most ichneumonids, we don't use them because they vary very little, but in some of these nocturnal groups, there's some lovely variation. This is an Absirtus. He has little spines on his Ediogus. Seems a bit unnecessary, but yeah. And then we've got Natalia, and these are 2 really similar species, Latungula and Cristata. Very different on the inside of their genitals. The pads are in different positions. Externally, they're really similar. This is just to illustrate my lockdown frustrations, trying to prepare without being able to make slides at home. I'm just trying to capture the shape of the pad and this wet specimen. That's the sort of outline of the pad. And then when it dries, it shrivels up and becomes a mess. So yeah, quite tricky sometimes to work with. You really need to ideally make slides or sketch or draw them or photograph them when they're very fresh and when you've got better equipment than I have. Just a quick, sorry, I realise I'm talking quite a long time now. Just to point out, I'm trying to collect as much data as possible on nocturnal species so we can start to understand change. This is my database. It's not massive yet, probably never will be, but since the 2000s, the recording effort has massively stepped up. So you can see the data flooding in, which is really, really cool. And there's a really enthusiastic bunch of people who are helping here. Are populations changing? This is something I'm really interested in exploring with some students. Hopefully we'll get some nice project out of this, but basically if you've got this This is Natalia cristata, very common. Here it is on a Dunbar caterpillar. They attack various hosts. But if you've got something like Natalia cristata, which is essentially a nondescript species that looks like many others, they're collected essentially randomly. So if you have all these nondescript specimens from a fairly random pool of nocturnal wasps, can you look at which species are relatively more common in different time periods and detect change? There's all of our total data set on the left, and there's the reduced data. There's the Natalia cristata data on the right, which is fairly similar. So it's broadly following the trend for all records. So maybe this is an averagely distributed species. Oh yeah, you can also look at blips, like when the National NCC was surveying various sites, so we got loads of data then. And there's my recording scheme. But here's Natalia miliorati, just 3 specimens known. And then suddenly in the last few years, I've been sent quite a few. Is that just recently spread a lot? Here's Caspialus adustus on the top, relatively much more frequently recorded in the distant past. Is this a species that has now become relatively much less common? And below is Caspialus cerebrator, which is the opposite. Very few old records, and now suddenly quite a lot of records. Perhaps that's following the fortunes of the small ranunculus, which is a favored host. And here's Caspialus repentinus, totally unknown until the 1980s, and now quite common. Yeah, well, not common. I've seen quite a few specimens now, and I've got a load more data to add to that. So again, can we detect change? And this is just to show this, just in the last 20 years, this is the percentage of increases in the number of species we know of in Britain of these 3 key genera. And most of this is just through increased knowledge, really scrutinizing the collections and improving our species definitions. And a lot of this, this increase in Natalia, 77% increase in species, is really for me going through the collections. So these old dusty drawers, all these species have been here all along in most cases. We're just discovering them now. It's the importance of collections, really. Although, as we've shown with some Caspialus, there are some colonists as well. So if you're going further with this group, do build a collection. It's vital. It's really important. Well, there's a lot you can do without having to take specimens, of course. But I think if you really want to make progress in the lesser known areas of ichneumanoids, you need to build a collection. Fortunately, with these things, you can get loads of specimens at light. It's quite easy to build up a long series. And there's often a bit of queasiness about taking lots of specimens, but one of the things about these, what can look like a sample of all one species can turn out to be about 10 or so different species. If you're preparing specimen, again, this is in the handbook, but one of the few good things about, one of the things that's quite easy about ichneumanoids compared to many other groups of insects is you just need to stick a pin through it if it's long enough, if it's large enough. You don't need to prepare them in any particular way. Maybe spread the antennae out so they don't interfere with the top of the pin. If they're small, I glue them onto a card point. Quite straightforward. Photos of live insects. Just to point out, I do get some really good data from from photos via iRecord and other methods. But if you're not really expert in the group, it's difficult to know what is identifiable and what isn't. I mean, these 2 photos, the one on the left, despite being a poorer quality image, is definitely Ophion scutularis, very skinny legs, quite dark color. The one on the right could be any one of about 5 different species of Ophion. So nothing more I can do with that. Maybe Slavoseki, as it was in September, but that's just a guess. Oh yeah, just wanted to point out, I put this on Twitter, but I was just really pleased. I found the species named after me in one of these samples from Herefordshire the other day. I think it's this Ophion broadi, described from Sweden 2 years ago. It should be in Britain. I think I've just found a specimen, so that was quite pleasing. But even here, although it looks like it, I can't be entirely sure. So I've got a leg being sent off for barcoding. I just wanted to quickly say, what is the future direction? Well, we're working at the moment on collecting. This is not quite as exciting as a drawer of wasps. It's a rack. It's a plate for barcoding with lots of legs. but these results will be invaluable. We're building up a DNA barcode reference library. So hopefully we can have every UK species, or most of them, with a verified DNA barcode. And I'm involved in this effort, as Leanna said at the beginning, this Darwin Tree of Life, which is trying to sequence the whole genomes of all the UK species. Early days for that, but at some point, all these Ophion and Natalia will actually have genomes behind them. We'll have their entire genetic code, which would be really exciting. But much more basically, there's still a huge amount we need to know about what are these wasps doing. We only have host records for about 2/3 of these nocturnal species, and even then it's just a few bits of data. We don't really know where they are. We don't know in detail when they fly or whether that is changing. So there's loads to find out. And obviously I'm hoping in this lockdown that the Natural History Museum are installing this massive Ophion in the central hall. I guess I'll find out when I manage to get back to work at some point. There you go. Right, that was a quick whiz through Ichneumanoids, which was a bit longer than I anticipated. Thank you for listening.
Thank you very much indeed, Gavin. That was excellent, and I hope you get your massive Ophion in the museum at some point. It looks like the future does sound really exciting, and even so much basic knowledge isn't known with this group that anyone really getting stuck in can really contribute and help make big strides. So it's a very exciting group. And one thing I would say is I just remember. Well, you said about getting stung several times by these wasps. I just remember quite a few years ago being near the top of Skiddaw, a third highest peak in England, doing some moth trapping, looking for northern dart, which we never found. But I just remember having seen lots of them in moth traps before, but actually being stung by one, I thought it was quite something. But maybe I'm just a bit of a wimp. But it was I remember it well.
Oh well, yeah, I suppose they are. I don't know. I suppose they probably are quite painful. I guess being a bit, as I collect quite a few social wasps as well, and I've got a bit used to really nasty hornet stings, so maybe I'm making a false comparison there. Yeah.
But that was brilliant, Gavin, and I think we'll go straight into some questions if that's all right.
Yeah, please do.
Okay, there's a few quite lengthy ones, so I'll try and be concise about this. So the question from Kath, over half an hour ago now, this is from the news yesterday, so maybe not particularly relevant to the nocturnal group we're talking about today. The news yesterday told of the National Trust having trouble with moth larvae on their wall hangings and tapestries. They got wasps in as a control. No names were mentioned. The announcer acted surprised as he thought visitors would be stung. The only quantification was the wasps were millimeters in size. Did the National Trust get in touch with you or anyone else for advice on this control?
No, not me. This this is a tiny little parasitoid wasp called Trichogramma evanescens, which attacks a range of insect eggs. So I guess within a confined environment, like indoors, it'll go for the closed moth eggs, as there won't be many other hosts. But out there, they attack all sorts of eggs. But so my colleague at the museum, Andy Polishek, has been involved in some of these projects to release tiny calcid wasps to control pests. But it's been explored for a few years now, this trichogramma. It seems to it's been used in various places around Europe. So I guess the fact that they managed to get quite a bit of publicity around this yesterday, which is all good for the wasp fans. But yeah, definitely not sort of what you would notice. This is small enough to develop entirely within a closed moth egg. So I mean, they're sub-millimeter things.
Right, and then I think there was a related question I think it's been answered now from Rahat Khan. Do we use these trichograms, parasitoids, against other moths or other pests? No, which you just said they attack other pests.
Yeah, and we use various. I mean, one of the classic ones, Encarsia, is another genus that attacks tiny little hosts. In this case, whiteflies, and that's something you can buy just to release in your greenhouse to get rid of whiteflies. There's a whole range of these. tiny little dust-like wasps, which you can use.
Okay, and then another question from Kath. So this is more general questions about this group, I think. So what do the adults feed on?
Oh yeah, good question. Their basic food is sort of nectar. So they will go to flowers, or it's thought that they probably eat honeydew quite a lot. But the other thing they eat, the females of some of these ichneumoniid wasps need to drink the hemolymph from hosts, or indeed non-host insects. So sometimes many species will jab a caterpillar and then drink the sort of hemolymph that oozes out, and they need that protein to develop their eggs. But other than that, they're basically sort of sugar feeders.
Okay, so can you attract them with sugar like you would with moths?
Oh, that's a good question. I've never tried. I've never tried sugaring actually, and nobody's ever told me that they've had success like that. It'd be good to know at night, because I've never seen these. No, tell a lie. I've seen a very few of the nocturnal species on bramble flowers in the daytime, but generally I don't see them, because I'm not out looking at flowers at night.
Yeah, they've never come to my sugar concoctions that I've that I've tried for moths. But
right, that's good to know.
Not that I've noticed. Okay, a question from Josie Hewitt. Just curious about the comment about how ichneumons don't or can't have multiple generations when presumably they parasitize some species of moth that are bivoltine. Is there any reason why parasitoids wouldn't follow the same breeding biology of their hosts?
Right. Oh yeah. So I should qualify that. That was really about Ophion, where as far as we know, Ophion species are univoltine. There may be exceptions, but some other groups are a bit more complicated. Like these Aliodes wasps can definitely be bivoltine or plurivoltine. In fact, some of these species, some Aliodes are really interesting in that they have 2 generations a year on entire, but their hosts are each univoltine. So they have to have 2 entirely separate hosts, which is weird. Conversely, a few of these Braconids have fast enough lifespans, developments, that they can have more than 1 generation on the same generation of their host caterpillars. It is a bit complicated. So, but Ophion seem to spend a lot of time in the cocoon for some reason.
Okay, so I'll go back to the questions. So this one from Steve McWilliam. Has the barcoding thrown up any issues with identification?
Yeah, I mean, so I must admit, so for the really interesting thing for Ophion is the fantastic sort of case of where barcoding has been incredibly useful, because one of the things that Nicholas was able to do in Sweden was get loads of specimens DNA barcoded, and then you look at your sort of clusters on the tree and start to think, does that make sense of this variation in morphology? Which is, so he had a sort of 2-pronged approach to sorting all these very similar things into different species. But it doesn't always work. So like Ophion Obscuratus is a group where so far the barcodes have not helped at all. There's no clear differences between groups which have different morphology and different hosts. And sometimes it's because they haven't, the mitochondria have not diverged. It's a recent split, so you've still got mitochondria that don't reflect the evolution of the wasps themselves. Or sometimes you've got these weird bacteria called Wolbachia, which you're sequencing instead. There's all sorts of reasons. And of course, it's just not infallible. It's a useful tool, definitely. But you've also got to be aware of contamination as well, of course. And some of the issues I've had in the lab have been to do with samples accidentally getting switched. And another, I mean, there's so many errors. A colleague who works in bryozoans was saying in the early days of DNA barcoding bryozoans, they had so many false results because what people were actually sequencing was all the things that live inside bryozoans or alongside them. So, got to be careful.
Would you expect when you have whole genomes sequences for all of these Ophions, would you expect things to start to become clear with that?
Yeah, well, yeah, I mean, you can then sequence a lot more genes, definitely. And one of the at the moment, it's unclear how you would use bioinformatically. It's really difficult to handle entire genome comparisons between lots of species because we don't have computing power is the limiting factor there. But one of the really cool things about having a whole genome is that you can then, if you're in the know, you can then find these gene regions which are. So Will was on the call earlier, who's expert in this. But basically, you can get all this sort of these whole genomes can help you then choose different gene regions to go back to older collections. So you may not be able to get long reads, good quality DNA out of old specimens, but if you have that scaffold genome for comparison, you can go back and sequence the relevant short bits from your collections. So you can go back and find loads more interesting genes that you could be sequencing. That's one advantage. But for me, one of the big things about having a whole genome is that hopefully we'll find out more about what they're, really interesting things like their venom chemistry, what genes are encoding for which venom proteins, peptides, what are the sort of, in those groups that have viruses, how are those evolving alongside them? And yeah, who knows?
Huge possibilities.
Yeah,
I'm just going to look, look again to the floor. Did anyone have any questions from the floor before I take another one from the chat? You can put your hand up now if you like. I can't see any.
There's
okay.
Ian Strachan had his hand up.
Hello, yeah. I just wondered if you're able to take specimens at the moment for confirmation of identifications.
Yeah, I can to a fairly limited extent. I'm encouraging people to send them to my home. So get in touch with me and I'll give you my home address, because I've literally been in the museum once in the last 3 months, I think. But yeah, I have been getting some interesting stuff sent to my home, actually. It's been quite fun.
Excellent. Okay. A question from Savannah Van Besdeg. I may be saying that wrong. I apologize. Would you be happy? Oh, it's the same question, really. Would you be happy to receive specimens? And if so, how would you like them to be sent to you and preserved? I don't know if these are coming from the UK or not.
Yeah, if it's a nocturnal wasp, I'm always happy to see them. But from the UK, okay. Yeah, I mean, basically, ideal is in ethanol or alcohol of some sort. But if they're dry, send them in a tube with a bit of tissue paper to stop them rattling around. They do get the antennae and legs easily break when they've dried a bit. But again, if they're dry and pinned, then that's fine, just as long as your package has loads of cushioning around it. Oh yeah, I should stress, I forgot to say that earlier, if you're identifying these things, always count the number of antennal segments before you do almost anything else, because it's so easy to knock the tip of the antennae off at any stage. So I always write it on the label, like number, I put F equals the number of flagella segments.
Good tip. Okay, a question from Dave B. Any ideas what the British Ophion Pteritis ag are? I have possibly 3 species keying out to this group.
Yeah, a lot of them will, I think in my limited experience so far, most of them are Ophion inclinans. There's also, I think it's called Arenarius, another species. Those 2 are the common ones I find in the Pteridis group. And there's probably another one like Eleni or Matti. I haven't looked into those ones yet. So yeah, inclinans probably the most common, but there are others, and there's lots to find out. And it's a difficult group, I find. That particular complex is the one I find most difficult.
Right. Any more questions from the floor before I take any more? No, I can't see any. One thing I was going to mention is just that the World Museum here does have a growing collection of these wasps. Thanks to one of the creators here, Tony Hunter. So once all the restrictions have been lifted and we are allowed researchers and people back in, then you are very welcome to come and use the collection. Any day of the working week. This is it's a national museum. It's funded centrally by taxpayers through the government. So please do use it. And I'm sure there's other sort of large collections in the country. Gavin, Naturalist Museum in Edinburgh, Oxford.
Yeah, there's several. I mean, Edinburgh has. I would say Edinburgh's probably got the best UK collection at the moment. Naturalist Museum's obviously pretty good, and visitors are always welcome if you want to come and see the national collection. But who knows when we're accepting visitors again? But hopefully not too long. Yeah, I'm sure. I've never actually visited Cardiff collection, but I believe it should be pretty good. But Liverpool's great, and obviously, Liverpool Museum is where I started really getting into parasitoids through a fantastically helpful bunch up there, like Steve Judd, Tom Maudsley, and others. Carl Klee, of course, Steve McWilliam up there. There's a great bunch of people. I'm really glad of Liverpool.
Yeah, all of those names are still in and out of the museum as well. So, okay, on to a question from Stephanie Skip. Is it known whether ichneumonids find their hosts by chemicals given off by the caterpillar itself or by the caterpillar's host plant? Could it be possible to make pheromone traps to see which wasps are attracted by which caterpillary smells?
Yeah, you definitely could. It's a really cool experiment to do, because what we know is mostly restricted to a few species of sort of economically important species. So some of these, for quite a few ichneumanoids, we sort of know that they're sort of attracted at longer range through sight. They're looking for particular visual cues, the right sort of vegetation and stuff. Then they tend to be homing in and detecting their hosts at short range. Quite often. So I'm trying to think. There's nothing that I know of for these nocturnal groups. Nobody's really looked at how they detect their hosts. Undoubtedly, they're recognizing them at very close range through their antennae, probably just touching them and getting some chemical cues off them. But it would be really, I'm just thinking now, cool projects you could do, but it would be really fun to know, because there are, of course, some wasps that are known to home in on feeding damage. The plants release particular chemicals when they're being eaten by caterpillars, which some wasps are known to home in on, or aphid damage. Again, the wasps can be attracted at longer range by the plant rather than the actual insect. Yeah,
they seem to have very large eyes for nocturnal group.
Yeah, I do suspect they're doing a lot of looking, getting a lot of visual hints, and because these things tend to be quite habitat specialist, I think they're often looking in particular niches, such as a certain height of vegetation, certain complexity of vegetation.
Right, yeah, a lot to learn. Okay, a question from Sajid Mir Entomology. Are ichneumon wasps acting as potential pollinators, and whether these parasitoids can be mass produced in the laboratory?
First, they're definitely involved in pollinate. Many species are involved in pollinating, but probably not particularly actively. They get bits of pollen on them, and they go to a lot of flowers, like probably a huge number of other insects in the world that are not generally thought of as pollinators. And yes, they can be various species have been mass reared in the lab. Things like control of this. The classic Ichneumonids work was on control of this sawfly, Neodipteron certifer, which is introduced to the North America, where they mass reared several species of European parasitoids to go and release over there. It does work. And some species of Pimpler, actually, not a nocturnal group, but Pimpler are quite common things that attack pupae of mostly moths. They are, they've been reared on artificial diets. You can, first, I think that's the first case, the first example where people have reared parasitoid wasps in entirely artificial media.
Okay, so a question from Pauline Pfeiffer. I hope I'm saying that right, Pauline, from the SMNS. Why don't you like the term parasitica, Gavin?
Oh, I'm just a snob. Because I suppose phylogenetically it's not a good term, because what it's doing is just, it used to be used as a suborder. People would divide up the order Hymenoptera into 3 suborders. And I didn't like that. I don't know, I was brought up as a sort of cladist, where we only give names to natural groupings, groups with one evolutionary origin. As parasitica was a catch-all phrase, but there's actually nothing wrong with it. It's describing a function rather than a group of wasps, I guess.
Fair enough. Okay, a question from Neil. Any news on updates to UK Natalia and/or Ophion keys?
Yeah, not a lot, because I'm slow. I'm very slow. But the Natalia, I'm trying to finish a paper at the moment. I basically bit off a chunk of work on Natalia, and I'm finishing that at the moment with Mark Shaw. I have quite a lot of barcode sequence data, which is helping. And what we're just going to do is sort out the group that really look most similar to each other, like that Natalia williamsi, which is this name I've had floating around for years. I'm going to publish that this year, hopefully. Ophion, no, it's really stalled because I need to go back through the collections, and I just haven't had a chance since, last March. And although I'll have loads of stuff sent to me from Herefordshire, from Peter Hall, which has been fantastic. But yeah, basically lockdown has not helped. So I'm biting off some easier chunks at the moment.
Okay, but it's something you're planning to do in the future.
Definitely, yeah.
Excellent. Another question from Pauline Pfeiffer. What do you think of the Sharkey paper published in Zookeys, in which he describes 403 new Braconids only via barcoding?
Yeah, that's been quite a controversial paper. Everyone in the world has quite strong opinions. Everyone in the taxonomy world. Personally, I think it's I don't know. In itself, it's a fairly terrible paper, is my opinion. But there's some grain of truth in there, which I suppose is that you can have a lot more condensed descriptions of new species, and you can rely a lot on barcodes. But I think they've gone out of their way to do things in the most sort of controversial way, almost. It's like, let's have purely barcode descriptions with really awful photos. That's not the future. You can easily have very good photos and some useful morphological descriptions as well. You could write a useful morphological description in the time it took to write the etymology sections of their new species descriptions. So a bunch of us are determined. I mean, they're onto something. It's not science. I don't think it's what they've done is science. They've just basically put names on some barcode sequences. That's not a species concept. That's just a sequence. So a bunch of us are trying to put together a paper which will also describe a whole load of new species from an area of the world where we desperately need to describe new species, but hopefully do it better in roughly the same period of time. Which is a bit of I don't know, perhaps I shouldn't, I'm not the world's best person for doing rapid taxonomy, as it takes me years to do anything, but maybe this is the shot in the arm that we need to actually get a move on and describe a lot more species.
Will those 400, will they sort of be correct? Is it reliable?
I mean, a lot of them will be, because there is very little described in those groups from Costa Rica already, but some of them I know will be synonyms. They disregard a couple of the genera they dealt with in that paper that I know fairly well, like Steropocitonus is one. I know they haven't bothered looking at any of the existing species, so I'm sure they've already re-described some of them. Yeah, that's a real shame.
Okay, we'll move on to a question from Catty. I think it's Catty, it could be Katie, but there's two T's. So I'm going with Catty. So what do caterpillars do to avoid being attacked, if anything?
Yeah, they're not defenseless, definitely. So small wasps can suffer quite badly, like these little microgastrines that lay their load of eggs in a big caterpillar. The caterpillars can bite them and chew them. Sometimes the wasp gets quite badly mauled. They do have defense, and obviously some caterpillars have defensive hairs and things, but really their best defense probably is in their immune system. Some of these Ophion are really only going for particular a sort of small range of species like the darts. Like Ophion slavisecki can probably develop in 2 or 3 species of Agrotis, but if it was to lay its egg in a wainscot, I imagine the egg would be encapsulated by the caterpillar immune system. Yeah, I mean, they do have pretty good immune systems. It's just there's a constant sort of war between the caterpillar and the wasp to overcome that immune system.
You see great images online of this, of sort of sawfly larvae rearing up in unison. Does that really help them?
I don't know. It's a good point. I mean, there's definitely, so it probably does help dissuade some more generalist species, but of course, some of these wasps have been co-evolving along with them for a long time, so they've evolved to counter these defensive measures. It's often unclear as to what they're defending against. Is it, sometimes it could be more generalist tachinid flies, for example, that they're trying to dissuade rather than particularly host-specific wasps.
Okay, another question from Robert Tracy. Do nocturnal ichneumonids pheromone mark hosts as mate finding would be an issue in the dark?
Yeah, good question. So they don't. What tends to happen? So the mating tends to be, as far as I can work out, probably up in the trees and probably quite soon after the female has emerged. She probably just pops out. Males can smell her, and then they'll mate. And then females only mate. A lot of ichneumonids actually, she'll only mate once or very few times, and that's it. But clearly, as to host marking, no, not that I know of. But what a wasp can often do to dissuade other, because obviously if a wasp is laying an egg inside an already parasitized host, then that can often be a waste of their own egg because it'll be lagging behind the original wasp. But what they can often do is basically stick their ovipositor in, and they can detect if it's already been parasitized, so they can avoid laying their own egg in. It's quite amazing how they do that. On the wasp mating, I would really love to know what's going on with Natalia, because they have these complex genitalia and these lovely brush-like claws. I imagine it's, for some reason, they're quite specialized in their external genitalia in a way that other wasps aren't, or at least other ichneumonids aren't. I've never seen them actually mating. Need to get them in culture.
Great. Okay, so a question from Santosh. I'm so sorry. It's a struggle. Is size of both eyes and ocelli relatively large, relatively bigger in nocturnal species compared to diurnal ones, or is it just eyes?
Both of them, yeah, tend to be pretty big. Yeah, the ocelli in many insects are tiny, and these nocturnal wasps, they're absolutely massive. Yeah, and both of them, there's probably loads more facets in the compound eyes as well.
Right. Okay, a question from Dave Skingsley. Are you aware if any of them a parasitizing hoverfly larvae. Just curious, as there is a hoverfly larval group, you might be able to ask.
Okay, yeah, so none of the nocturnals do, but there are hoverflies have quite an interesting fauna of parasitoid wasps that attack them. So in Ichinomonids, there's a specialist subfamily called the Diplazantini or Diplazontini. I never know how to pronounce these things. So there's a whole little subfamily which specialize in hoverflies, and they lay into either the eggs or the larvae and then emerge from the host puparium. So yeah, really cool. There's also a bunch of chalcids and some phagitids that attack hoverflies. They seem to be quite a popular group for parasitoids. Nice and juicy.
Do you get sent anything from hoverfly breeders?
Not a lot. Somebody had
rearing.
Yeah, Judy Webb sent me some interesting things she'd been rearing out. It's this genus called Rembobius. It's a cryptine. Yeah, I don't get much actually. One of the interesting things about the hoverfly parasitoids is that there's quite a lot of host records for hoverflies that feed on aphids, but for other groups, there's very few host records. It may be they're just not attacked by many wasps, or it could be they've been neglected, and maybe there's some really interesting ones attacking them.
I know someone in the Northwest who does rear hoverflies. I'll ask them what if anything is coming out.
Yeah, that'd be good.
I'd be interested to know that. That'd be fascinating to find those.
Okay. Yeah, there is one really interesting little genus of Diplazantine, Bioblapsis. They've been reared from Ferdinandia in rot holes, which is quite different. So maybe some of these other interesting weird Diplazantines are also going for hoverflies in weird situations. Yeah, it'd be really cool to know.
Okay, we'll go to a question from Graham Smith. Is there a reason that so many nocturnal species have a rather orange coloration?
Oh yeah, yeah, we touched on that earlier. It's just they just don't. I think they don't need color patterns because they're flying around in the dark, and they're presumably making your cuticle dark. You know, as many of these wasps have a ground color of black or dark brown, and that's just. There's no point making all that melanin, which is energetically costly if you're flying around at night, would be my guess.
Sounds very logical. So is any other questions from the floor? We seem to be out of questions from the chat now.
Can I ask a question on it? It just occurred to me because I work with some tropical species that get sent to me that have yellowish patching on them, and when I use a UV light on them, it comes back with a UV reflectance.
Okay.
I'm not sure if anyone's had a sort of wafted a UV pen over them.
No, although I have. No, that's a good point. No, I don't. I've never seen them fluoresce with UV lights because I have been out at night with the UV lights, but I'll have a go. Yeah, that's a project for the this year.
Oh, okay. A couple of questions coming in now. A question from Jazz B. Where do the Ophionini adults roost during the day, and are any species active during the day, and are any reared host records for the Ophion Obscuratus aggregate?
So to answer that, where they roost during the day is really speculative, but when I've noted, often they rest. Ophionine adults rest with the wings sort of flat and their abdomen sticking up, which is a weird posture. And because when I release them in the day, they often seem to fly up into the trees. I suspect they rest on the undersides of leaves a lot of the time, maybe close to the petiole. It's a total guess. I never find. I don't think I've ever seen them roosting. If anybody has, that'd be great to know. My assumption is they're on the undersides of leaves. But some species are active in the day, but often that's males. So males of things like Ophion Ventricosis and Eromatilis marginatus, you can see them flying around very actively in the daytime. They basically I presume they're out searching, trying to get a head start on finding the females before they emerge from their cocoons. I've seen massive aggregation of Azeli albiditarsus males as well around a tree, presumably waiting for the females to pop out. And any host records, the Ophion Obscuratus aggregate, yes. Some of them attack things like wainscot larvae and Zestia larvae. Some attack heathland. So one of the, I can't remember now whether it's the Sylvestris or autumnalis, I guess with their that attack least yellow underwing and true lovers not. So they're probably partitioning up, they probably go for different groups of noctuid moths that are active at those different times of year, I guess.
Okay, over a question from Simon. Can we rear parasite larvae found on spiders? Well, Tony brings them in occasionally with these spiders with larvae on their sort of neck area, I would say, in a nice unscientific way. But yeah, it's quite yeah. So gruesome, I suppose. Gruesome way to parasitize.
Spider parasitoids are great to rear because they're often really easy. You just keep them in a tube and make sure they're clean and give them some flies. Yeah, really nice. And watching them, because again, they're ectoparasitoids, so watching them going about their business is quite fascinating.
Any of those in the nocturnal group? No.
Not habitually nocturnal, no. Some of them turn up at light traps quite regularly, but they don't look like they should be nocturnal.
Okay, a question from Annie Smith. Do the males and females have different life expectancies?
Yeah, well, again, for these particular nocturnal groups, we don't know. But for many wasps, yes, males live shorter lives. Females tend to, I mean, they're just often males are smaller and just, I guess, less durable. They're not as, yeah, they're just not as useful, I suppose. So they tend to have quite short lifespans. But there's not a lot in it. I mean, females can exhaust themselves by, the process of handling lots of hosts as well.
Tony Hunter was just commenting on the roosting thing. I think he found an Ophion on the underside of a spruce branch a few years ago.
Okay.
I can't remember which species that was.
Oh, nice.
Also, a question from Jazz B. Ophion in the larvae are endoparasitoids. What happens to the larval feces? Generally, do Hymenoptera retain feces in the gut until they have completed feeding and pupation?
Yeah, I mean, as I think Pauline answered that in the chat as well. It's just, that's a really neat feature of Hymenoptera, other than, so all the sawflies have the normal sort of, eating and defecating, but the other groups which evolve, so all the parasitoids and the aculeates have a common ancestor, and they don't have the larvae don't have a gut that connects. So basically, yeah, they have to store up all the meconium until when they're just about to pupate, the gut, the hind gut connects to the midgut, and then they do a massive defecation, which I'm sure is a huge relief.
Right, and Jennifer Jones just said that she'd found an Ophion in a hedge during the day. It didn't seem very active, so that lines up with leaf undersides. So from Annie Smith. Oh no, sorry, I've already said that one. A question from Steve McWilliam. I found them in rotten wood in winter. Will they be overwintering, or will they have hatched out from beetle pupa? Yeah,
there's a bunch of, so there's a subfamily Ichneumonidae, have loads of species that winter as adults, and they'll often overwinter in rotten wood or sometimes in grass tussocks. Some species go into caves. Yeah, they really specialize in it. In other subfamilies, there's only a few species that seem to do that. And obviously Ophion Obscuratus, or one of that group, whichever one is out now, must spend quite a lot of time in the winter just being still somewhere, waiting for the nice weather for the caterpillars to come out. Yeah, this it's only the female, those hibernating groups, it's only females that do it. The males die out in the autumn.
Right, okay. A question from Finley H. Will each individual wasp lay in several larvae, or does each wasp only parasitize one individual larva?
No, they've generally got, so a female ichneummonid, it very much depend on which group they are. Some of them will have, say like, I suppose the lowest numbers are about 20 eggs, but some have a potential fecundity of thousands. They can generate more eggs. So it varies very much depending on their life history. But they'll definitely have a whole bunch of eggs that need to be laid. But obviously all these, so most of the ichneumonids are solitary. They'll lay one egg in one caterpillar. They'll obviously then go off to try and find more caterpillars. Whereas braconids have more gregarious species, which will lay a whole clutch of eggs in a caterpillar.
Okay, a question from Augustine. I can't see your whole name. D. Ketel something. I'm sorry, it does not. The whole name's not coming up on my screen. But you are asking: are there many wasps that happen to be caught on light that are not nocturnal, or is it likely that they are also partially nocturnal?
Yeah, there's loads. So I used to go through all the bycatch of this of Rothamsted light trap, which is at Monk's Wood, where I used to work. And over the 3 years that I was doing that, I mean, I got loads of species at that light. And some of these were quite regular, but they look to all intents and purposes like totally diurnal species. So I'm guessing they're either still active quite late in the evening when the light comes on, or they just continue, and maybe it doesn't matter to some of them whether it's day or night. Yeah, you can get quite a variety. It's just the point about my recording scheme for nocturnal ichneumonids is to try and concentrate on those groups that are recognizably nocturnal, because it narrows the focus a lot.
Okay, a question from Fons. Are there any nocturnal spider parasitoids, for example, Pyogaster, which we once caught at light?
Oh, yeah. So I don't know about Pyogaster because they're so rarely caught. They're quite pale, so it could be they're nocturnal. I've had a bunch of these polysphynktines, the spider parasitoids, or one of the most well-known group of spider parasitoids. I used to catch quite a few of those at light, so I'm guessing they're often active into the night. But none of them look particularly nocturnal.
Okay, a question from Cough Nod Richard. Are spider parasitoids species specific to their host? And if not, do the wasps vary in size depending on the host?
So as far as so obviously it varies a bit between species. Some of them are very, as far as you know, some of them are pretty specific. What they tend to do, I'm probably grossly misrepresenting it, but I think a lot of the spider parasitoids tend to attack a narrow range of hosts, so closely related species in a particular niche. And yes, some of those that do have a range of hosts can vary a lot in their size. And that's true of any of the parasitoids that are quite generalist can be enormously variable.
Yeah, in the collections in Liverpool, I mean, opening the drawers, things that look like it can't possibly be the same species. This difference in size is so huge. We'll go to a question from the floor now. Ian, you've got your hand up. Would you like to? Sorry if you've been waiting a little while.
No, not still. That's fine. I just wanted to ask about the records that you have. There seem to be virtually no records on the NBN in Scotland anyway. And are you submitting them, or are you doing distribution maps yourself?
No, that's a very good question. So I've put off submitting them so far because the taxonomy keeps changing. And I know from experience of working with the UK species inventory at the museum that, the checklist sort of underpins a lot of the databases out there, but also I'm just a bit slack. So for the first bunch, like Caspialus, I did the distribution maps myself, and that data should have gone to BRC, and I should check whether I actually sent it there or not. For other groups, yeah, I mean, when people have requested data, sometimes I've just given it to them because I obviously have no problem. So if record centers have requested, if I have anything, and I've just passed it on. But I know I've got a lot of catching up to do. So things like Caspialus could just be passed on now. There's no reason other than my tardiness. Ophion needs loads of cleaning up before I could pass that data on anywhere. And Natalia needs me to finish a couple of papers so that some of these names are actually validated. Yeah, it's complicated. The intention certainly is for them to be publicly available, and anything that's been databased to a species level through the NHM collections, Natural History Museum, any of that data gets fed through to GBIF.
All right. Okay. Thank you.
Okay, is there any more questions from the floor or I can't see anything in the chat. Can't see anybody's hand up. So you may have overcome all of these questions and exhausted everybody now.
It's been a fantastic bunch of questions.
Yeah, it's
I see Rod's got his hand up.
Oh, sorry, Rod. Do you want to unmute yourself, Rod?
Okay. Do the humanoids themselves have parasitoids?
Yeah. So when they're inside the hosts, yeah, they can be attacked by mesocorines, which is a specialized subfamily. There's the odd other true hyperparasitoid as well, which specialize in the humanoids inside their hosts. But then often when they've popped out and pupated, they're then vulnerable to other parasitoids. So they're not true. So in that case, you know, the parasitoid that parasitizes an ichneumanoid cocoon is not acting like a true hyperparasitoid because it's not going inside another host. You know, it's not having to go inside a caterpillar to find its host, but still, in terms of its population cycle, yeah, it's acting like a hyperparasitoid. All these, there's a weird case I know of. This little calcid wasp, not my group at all, but there's a few species which have a weird life history where a female will lay a male egg inside the larva of other females' offspring. So the males develop as hyperparasitoids of their own species, whereas the females don't.
Where does it end?
Yeah. The record that I'm sure there's, I think it's the record is something like 7 layers of parasitism within an aphid. You know, one of the amazing things is how incredibly efficient they are at eating their host, because they're converting almost all of the available host into their own body tissue.
Yes, we had a caterpillar at the back here. Can't remember the species, but it disappeared, and all it was left was a group of white fluffy eggs, which another small tiny wasp was parasitizing.
Yeah, yeah. There's no escape from wasps, even if you're a wasp.
And is there presumably the bigger the caterpillars, the eggers and the hawk moths, presumably you can get bigger sort of chains of ecosystems of wasps within those? Does it work like that?
No, well, you'd have thought so, but it doesn't quite, because what tends to happen is the wasps that attack the hawk moths, or at least the Braconid wasps that attack the hawk moths, tend to arrest their development before the hawk moth gets very big. Quite weird. Some of the Ichinomonids attack full-grown hawk moth caterpillars, so they're pretty impressive beasts. But they don't have any hyperparasitoids as far as I'm aware. They're probably too big.
Right. Yeah, I think we've got to the point where there's no more questions outstanding again. So I think, oh, here we go. So we have another question from Augustine. I have a specific question about Abistritus specimens. They often have characters of both species. Is it possible that they are just very variable?
So Abscertus, yeah.
Sorry.
That's right. So in my experience, although they are variable in their characters, but I've always found it possible to distinguish two species, at least in Britain, Vernalis and Vicinator. One on the sort of, if you got them side by side, Vicinator is a sort of more slender insect, and it has a particular shape to the petiola area, for those who are really interested in this, whereas Vernalis has a broader petiola area, and it's kind of a stockier insect. So there are differences in the Propodium I find most useful. The wing characters do vary a bit. So, I mean, there could be, so yeah, there may be some species that are more tricky, some specimens that are more tricky, or it could be there's also another species in the continent, which I haven't seen in Britain. Yeah, I'd have to look at more.
Yeah, I'm not sure where their specimens are from, but likely looks like.
I think it's Belgium.
Belgium, right?
I'm not sure. Anyway, yeah, I'll try and, because I published some provisional key for separating them, so maybe I need to revisit that.
Okay, a question from Steve Orridge: does neoteny occur in Ichneumonids?
I can't remember what's meant by neoteny.
Neoteny. Yeah. Yeah, no, I don't know.
It's the juvenile state. They don't reach adult. They don't or the larvae develop genitalia but don't develop wings, legs, or whatever.
No. So no, the closest that I know of is a few parasitoid species where it's known. So they're gregarious, and you have larvae which will eat, because you've got gregarious species inside a host caterpillar, and then in a few species, it's known that some of the larvae don't develop into adults. They instead basically stay behind to kill off any other endoparasitoids inside the host. They act like guards and soldiers. It's the only example I know of in wasps where they don't develop to adulthood deliberately.
Thank you.
Simon asked about gynandromorphs. Yes, I published a little note with Mark Shaw on 2 gynandromorphic pneumonids that I'd noticed. Exciting paper, which I expect never to be cited, but they were quite interesting gynandromorphs in that they were anterior posterior, so they could only be detected because these were species in which the head end, the anterior of the females looks quite different to the anterior of the males, and that's probably a bit more common than we imagine because of the way that embryogenesis occurs in some wasps. But I've never seen a bilateral gynandromorph wasp, though I have seen a couple of specimens of genitalia of both male and female, which is weird.
Well, I think we'll leave it there then. But thank you so much, Gavin. That was and such incredible knowledge to be able to answer all of those questions. And it's great that you'd be able to. The consequence of this pandemic is that instead of just speaking to 15 people in the museum or whatever it was when you came in, we've had 200 people here that you'd be able to share your knowledge with and inspire people hopefully to start looking at these closer, especially in the moth traps. I know there's a lot of lepidopterists that have come to this meeting today, so hopefully they'll give these things a bit more a closer look.